In cells, like these neurons, proteins are constantly synthesized, and any misfolding could lead to protein aggregation. The persistence of non-functional protein aggregates in neurons has been associated with many diseases. Proteins are molecular workhorses of the cell. They have many roles, including the assembly of intracellular structural support and molecular motors, or the catalysis of biochemical reactions. The functions of proteins depend on their specific three-dimensional folding and stability in the cellular environment. Proteins occupy up to 40% of the cytoplasmic volume, creating a molecular crowding that increases the risk of protein aggregation between hydrophobic patches in unfolded regions of proteins. Targeted degradation by proteasomes normally limits the consequences of protein aggregation. Not all proteins diffuse freely. Vesicles transport to their final destination secreted proteins, membrane proteins, and proteins targeted to intracellular organelles. For example, vesicles destined to late endosomes are formed at the surface of Golgi stacks. With the help of clathrin molecules that assemble into a coat, promoting the curvature of the Golgi membrane. Shortly after their release, the vesicles shed their clathrin coat and adaptor proteins. Synaptic vesicles undertake a long journey along axons. To overcome limited diffusion caused by molecular crowding, motor proteins transport vesicles on long fibrous proteins called microtubules. Few kinesin motors move each synaptic vesicle toward the plus end of microtubules. Free kinesin adopts an inactive folded conformation. Upon binding with the vesicle, the kinesin stalk region unfolds, and the two kinesin heads are free to interact with the microtubule. The ATP-dependent switch between microtubule-bound and free states leads to the alternating swing of the heads that characterizes the hand-over-hand walk of kinesin. Conformational changes and electrostatic steering promote unidirectional movements of vesicles. At any given time, one kinesin head is attached to a microtubule, thus favoring long-range uninterrupted vesicle transport. If a vesicle detaches, its very limited diffusion increases the probability for fast reattachment. In the crowded environment of the cell, mutations altering protein folding increase the risk of aggregate accumulations, which play a key role in the pathogenesis of many neurodegenerative diseases.